So what exactly is gene therapy? It's the addition of new genes to a person's cells to replace faulty genes in an attempt to prevent, improve, or cure disease. Genes need a bit of help to get into cells. One way they get that help is through a virus. Now, think about what viruses normally do. They are really good at getting into our bodies and making us sick. They do that by injecting their genetic material into our cells. So scientists had the great idea of hijacking a virus to deliver genes into cells. But don't worry, they remove the parts that make us sick. The modified virus is called a vector.
History of gene therapy. In 1970s and earlier, Friedman and Roblin authored a paper in Science titled Gene Therapy for Human Genetic Disease. Rogers was cited for proposing that exogenous good DNA be used to replace the defective DNA in those who suffer from genetic defects. In 1980s, a retrovirus vector system was designed that could efficiently insert foreign genes into mammalian chromosomes. In 1990, the first approved gene therapy clinical research in the US took place on 14 September 1990 at the NIH under the direction of William French Anderson. 4 years old, Ashanti De Silva received treatment for a genetic defect that left her with a severe immune system deficiency. In the year 2000, the modified cancer gene therapy strategy of antisense using triple helix anti-IGFI approach was registered in 2002 by Wiley Gene Therapy Clinical Trial. And finally, in 2017, in March, French scientists reported on clinical research of gene therapy to treat sickle cell disease. Cell cure in 2017. In March, researchers announced that a teenage boy in France had been cured of sickle cell disease after receiving an experimental gene therapy developed by Bluebird Bio. So how do scientists do? The scientists removed stem cells from the boy's bone marrow and modified them in the lab by introducing copies of a gene to prevent his red blood cells from becoming sickle. When the treated cells were infused back into his body, they began to make normal red blood cells. DMD is the most common hereditary muscular disease among children. The researchers have developed a gene therapy that may provide permanent relief for those suffering from DMD. So how do researchers do?
People with Duchenne do not have a working copy of the Duchenne gene. The hope is to deliver a new working gene into muscle cells using a virus. The virus's goal is to carry the new Duchenne gene to cells throughout the body. The Duchenne gene is big. Researchers made a shorter version to fit into the virus. This shorter gene has the vital parts, but it isn't as good as the whole gene, so it should provide benefit but is not a cure. Researchers expect muscle, heart, and breathing benefits for most people with Duchenne. It should work better for people with more working muscle cells because gene therapy will have more difficulty getting into damaged cells.